Okay, thank you. Thank you very much. So I'm very happy to be after this very great presentation because you already said a lot of things that are very useful for me because a lot of introduction has been done. I will try to give an overview of the idea of Secretome. That is something quite new, but as we will see with this history, I will try to make it's a concept that has been developed few years ago. So I would like to start from one of the first publication about mesenchymal stem cells, actually the first one, and it's 50 years ago. And about the MSC revolution, as Professor Kaplan said, MSC is not anymore mesenchymal stem cells, but 50 years ago, this was really the idea of mesenchymal stem stromal cells, so of cells that can do something. And now we know that mesenchymal stem stromal cells or medicinal signaling cells, as we want to call this kind of cells, have a perivascular origin and maybe a subset are actually pericytes. This was the idea of cells differentiating into something else that really lasted for many, many years, let's say 70s, 80s, and probably until also in the 90s. But at the beginning of 2000, it became clear that mesenchymal stem stromal cells, or MSCs as we want to call them, are able not only to differentiate, but also to release something. And in this publication, it's one of the first publication with this concept. It's interesting to read in the last sentence that the cells may function as supporting cells. So this was the first idea, the first concept that MSCs may be cytokine factors. It is important to say cytokine because for many years the word cytokine or chemokine or soluble factors has been the word or the words to describe what is released by MSCs. And this is actually what also has been explained in the next few years. In this case, in 2005, it became clear that mesenchymal stem cells or MSCs may secrete a lot of factors, a lot of cytokines that, for example, can be supporting for hematopoiesis. And this allowed the idea that the secretive factors can be very important also for therapy, because not only at the time the idea was to use cells to replace damaged tissues or to have a new tissue, but also it started to appear the idea that these cells can also support the environment where they are. And this was an example, and another example in 2006, where it was, for example, rheumatoid arthritis, in one case chondrocytes, in another case T cells, so that the factors released by MSC may modify the surrounding environment and interacting cells. Not only the old idea that they differentiate, and now we know this idea is probably wrong, or at least only partial true, that they can differentiate to something else. So this opened the door to a new concept and a new player, the concept of the conditioned medium that now we will see more modern idea of the secretome. So the conditioned medium was all this medium you can collect from a petri dish where you seeded MSCs. And the conditioned medium was thought to have a therapeutic option, to a therapeutic potential, and both for regeneration and also, as you can see in the second publication, in 2007, so it's already 15 years ago, to be used for the treatment of inflammatory condition. So it started to appear the idea that not only the cells, but also what they secrete may be useful for a therapeutic option and for to heal and to promote something. And then we went to 2011 with one of the most famous publications in the field from Professor Kaplan and Dr. Correa about SC as an injury drugstore. And this is the first, really, I think the first clear idea that MSCs are an injury drugstore, so are medicinal signaling cells, and then that they can produce something, and this something now can be called the secretome, both if the cells are in the environment or are injected for example, in a pathologic joint, or if they are in a petri dish. So all the factors that can be secreted by the cells and factors that can interact with targets. And the secretome, MSC secretome. In my opinion, it can be seen as, and this is also the title of this presentation, as an orchestra with a lot of different players. All these different players may have a different influence on the target cells and really are important for the function of MSCs. Historically, the most studied players of this orchestra are soluble factors, so cytokines, chemokines. There is a old picture of a paper, but it's still quite actual. We see that these factors have really a lot of function. There are a lot of anti-function, anti-apoptosis, anti-fibrosis, antibacterial, as Professor Kaplan and others said, but also they have also pro activities. So they can stimulate proliferation, they can have an immunomodulatory function, chemoattraction, support for different processes. And there are a lot of evidences that these factors in the field of the joint disease, of course, there is a lot of evidences that these secreted factors can have a healing activity, both on the tissues and also on the immune cells. So they can promote the cartilage regeneration, the cartilage restoration, and they can also promote a switch of all the inflammatory activated cells from pro-inflammatory to anti-inflammatory, in particular macrophages from M1 to M2. And then the second player of the orchestra, the extracellular vesicles. Extracellular vesicles became quite popular in the last year. We know that our vesicles secreted from the inside of the cells, like exosomes. So the word exosome is very popular, and usually it's used maybe not really in the proper way. But let's say exosomes are only vesicles secreted from the inside of the cells, and then microvesicles that are secreted from the surface of the cell. And this is a quite new concept. It's a quite cool concept. Everybody likes the word exosome and the word extracellular vesicles. But it's something new only because now it's popular. Because the first notice of extracellular vesicle was in 1966, talking about platelet dust. And this dust released from platelets at the time was really considered as dust, as garbage. But now we know that these vesicles are not really garbage, but contain a lot of factors, a lot of molecules, and most importantly, microRNAs. And we know that microRNAs are able to interact with the cells from the inside, modulating the stability and the translation of a messenger RNA. And in this perspective, in the field of joint diseases, some in vitro studies showed that the exosome, the purified exosome from MSCs, have a really interesting potential. Again, in this case, for tissue regeneration, tissue homeostasis, and immune cell switch from pro-inflammatory to anti-inflammatory status. So as a whole, the secretome, so composed of cytokines, chemokines, and extracellular vesicle, may act in the joint disease on different levels. So we have the cytokines, chemokines that can act directly on the surface of the cells, of the target cells, with the interaction with the receptors. And the other way, the microvesicle may interact with target cells by being incorporated in the cells and release microRNAs and other factors that can modulate the target cells from the inside. And this double activity from the surface and from the inside has been shown altogether to lead to chondrocyte proliferation, to reduce apoptosis, fibrosis, hypertrophy, inflammation, and all these features may lead to cartilage homeostasis restoration and also reduction of inflammation. But there is a point that is crucial about the secretome. The point is that when we talk about cells, and especially we have seen in this previous presentation that cells may interact with an inflamed joint, these cells may, by interacting, modulate their activity. So they also can modulate their secretome. If we think about secretome as a per se therapeutic product, so produced in a bio-GMP facility, it's clear that the secretome has a big lack with respect to cells. The secretome cannot modulate itself. If a cell can modulate its activity and interact with the environment, the secretome is what we produce. So in the last year, it became clear that if you want to have a secretome with even higher capacity on the target cells and the target tissue, we have to, this secretome has to be produced, let's say, in a clean room, so let's say in vitro, we have to modify the producing cells in order to have a more active secretome. And in the last years, my group and other groups really work on this concept, and it has been demonstrated that as Professor Kaplan said, that MSC can be pro or anti-inflammatory. If we modify the in vitro environment, we can produce different types of secretome. And for example, if you have a high level of inflammatory mediators, like interferon gamma or interleukin-1 beta, we can have a secretome that is modified in its composition and is really even more able to allow a cartilage or chondrocyte homeostasis, regeneration, proliferation, and immune cell modulation. And especially for the exosomes, so this part of the secretome, it has been demonstrated in several preclinical models, so in animal models, that there is really a benefit on the tissues, and especially if in exosomes, and the idea is also with for the secretome that has been modified by preconditioning the cells, the results are even better. So we can say that the secretome can be taught as a therapeutic option per se, and has a very good potential. But if we can modify the producing cells, we can have an even more active secretome. So we can somehow resemble the interaction mesenchymal stem cells on MSCs have on the in the real environment. We can reproduce this environment and then collect only the secretome with the advantage of having a cell-free product. And if we have a look at clinicaltrial.gov, this was done a few days ago. At the moment, there are 5-6 clinical trials working with the MSC secretome. At the moment, there is not any secretome approved by European agencies or the FDA, but there are some clinical trials. And actually, out of 5 at the moment, 2 are related with joint diseases. So this is quite interesting for our field. So, in conclusion, we can say that the secretome have several components that can modify target cells and tissues. Secretome intended as the secretome of the living cells in a tissue, where we inject them for example, in a disease joint, but also secretome intended as a per se therapeutic product, as a clinical product. The most promising features are tissue regenerative and immune modulators. So we can see really an effect on, for example, on cartilage, on synovia, and on immune cells, especially macrophages and T cells. And these features are the basis for a potential regenerative medicine application in several fields, and of course, in the orthopedic field. And thanking you for your attention. Just mentioned my in my lab, and thank you very much. And I hope that I gave you an overview of the secretome and its potential in the clinical application, especially in the frame of orthopedics. Thank you very much.